Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, in the previous classes, we have studied about the anatomy of dicot and monocot root, dicot and monocot stem, and in this lecture, we are going to study the anatomy of dicot and monocot leaves. Dear student, as we studied in the ground tissue system when we are talking about that, we have already talked that in leaves the mesophyll cell are going to represent the ground tissue. Means the ground tissue is not going to represented by so many layers or so many different type of tissue layers which are differentiated, but here it is going to be represented by a mesophyll cell. So let's discuss dicot and monocot leaves anatomy. So when we see the dicot leaf, which is also known as a dorsiventral leaf, this dorsiventral leaf, if we talk, this dorsiventral leaf, if we talk, and this is the dicot leaf. When we talk of transverse section of this dorsiventral or dicot leaf through the lamina, show three main parts. When we take a cross section or transverse section of this lamina, the three main parts are seen. That is mainly epidermis, then a mesophyll cell, and you will see the presence of vascular bundles. These three are the main parts which are seen in this particular region. Okay, so if we see the transverse section look like this of specifically dicot leaf. Okay, now you can see in this particular transverse section of dicot leaf that here the bundle sheath are present here, and here you will see the presence of xylem, you will see the presence of phloem, means vascular bundle. This is the epidermis, this is palisade mesophyll, this is air cavities, this is mesophyll, spongy mesophyll, this is substomatal cavity, this is abaxial epidermis, and this is a stoma. So dear student, if we talk here in dorsiventral leaf, if we take a transverse section, then you will see the adaxial epidermis. This adaxial epidermis is nothing but a upper epidermis. So upper epidermis is also known as adaxial epidermis. Then if we talk about abaxial epidermis, this is lower epidermis. So this lower epidermis is known as abaxial epidermis. And between the upper and lower epidermis, you will see the presence of mesophyll cell. But the mesophyll cell in dicot leaf are differentiated into two types. That is a palisade mesophyll and the spongy mesophyll. You can see this palisade mesophyll are somewhat columnar, okay, whereas this spongy mesophyll are somewhat oval or rounded, you can say. So here in palisade mesophyll, when you see they are compactly arranged, and they are compactly arranged, so in that the intercellular spaces are is absent in palisade mesophyll. But if we talk about the spongy mesophyll, then in spongy mesophyll you will see the presence of air cavity or intercellular spaces due to their shape. Okay, now on the lower epidermis, when we talk about dorsiventral leaf, the number of stomata on lower epidermis are more than the upper epidermis. So here there is a no space in the palisade mesophyll, but space are found on spongy mesophyll. Then yes, the number of stomata are more on the lower epidermis, okay, on the abaxial epidermis compared to the upper epidermis. So there is a what a difference in the dorsal and ventral surfaces in the number of stomata. Now we remember the number of stomata are more on the abaxial epidermis. In this abaxial epidermis is also known as dorsal epidermis. Or why? Because this is present on the dorsal surface. Whereas adaxial epidermis is also known as the ventral epidermis because it is present on the ventral surface. Now, here, apart from this, when a stoma is present, below the stomas, a small cavity is present which is known as substomatal cavity. And when we see this structure, here you will see the presence of this bundle sheath cell and a layer of bundle sheath cell. Then you will see the xylem and phloem, that is a vascular bundle which are present here. Okay, and below that you will find this particular structures which are known as bundle sheath extension. Bundle sheath extension are seen here. Okay, so this way you can see the particularly presence of all particular uh, diagrammatic representation and all the labeling. Now we have to understand this one by one, and we have to note some of the important points with respect to this dicot leaf. Then, if we talk the dorsiventral leaf, and when we talk about this dorsiventral leaf, they are also known as bifacial leaves. And in this dorsiventral leaf or bifacial leaf, dear student, when they are present at a right angle to the stem and you see the particular transaction, you will see that why they are known as dorsiventral leaf, because the first thing that in this the stomata are more on lower surface, on lower surface than the more on the lower surface, and on upper surface they are less or absent. Okay, now after this, if we talk, the upper and lower epidermis are there. Just we have seen that upper epidermis, it is known as adaxial epidermis or ventral epidermis, and the lower epidermis is also known as the dorsal epidermis, or we can say it is a abaxial epidermis. Now, when you see both the epidermis, you will see that cuticle is present on both surfaces. On both surfaces, but cuticle on upper surface is thick. Cuticle on upper surface is thick, or we can say a more thick. Now, when we see the upper and lower epidermis, then between upper and lower epidermis, mesophyll cell are present, and this mesophyll cell, we can say, represent ground tissue in leaf. Okay, so this is the mesophyll cell which are present. But when we see the mesophyll cell in this dicot leaf or bifacial leaf in a transverse section, these mesophyll cells are differentiated. So here this mesophyll cell are differentiated into two type, and this two type are, one is palisade mesophyll and second are spongy mesophyll. When we see in the diagram, this palisade mesophyll are also known as palisade parenchyma because they are made up of parenchymatous cell, whereas spongy mesophyll are all made up of parenchyma as well, so they are known as spongy parenchyma. Now, when we talk about this palisade mesophyll, these are situated towards upper epidermis or adaxial epidermis or ventral surface, and it is made up of elongated cells. It is made up of elongated cells and which are vertically arranged parallel to each other. And these are vertically arranged and have more chloroplasts, have more chloroplast and large vacuole. But when we see the important feature due to the elongated nature in palisade mesophyll, the air cavity are absent or intercellular spaces are absent. But when we talk about another type, that is a spongy mesophyll or spongy parenchyma, in that, dear student, if you talk, the spongy tissue or spongy parenchyma are situated towards lower epidermis. They are situated towards lower epidermis. And when they are situated on the lower epidermis or abaxial or dorsal surface, their cells are oval, their cells are oval or rounded, and due to the oval or rounded nature, the large air spaces or intercellular spaces or air cavity are present. Okay, so this is the what actually anatomy of dicot leaf. In dicot leaf, dear student, this is about the mesophyll cell. But within the mesophyll cell, you will see the presence of vascular bundles. So when we talk about that, dear student, When we talk about the vascular bundle, specifically this vascular bundle in leaf, not dicot, in both dicot and monocot leaf, the vascular bundle are conjoint, collateral, and closed. Closed means vascular cambium is absent. Now here, when vascular bundle are absent, in case of this, cambium is absent in this vascular bundle. Actually, not vascular bundle, cambium is absent. So secondary growth does not take place. But apart from this, you will see the two distinct patches of parenchyma, mainly or collenchyma, are present above and below the large vascular bundle. Okay, and this particular cells of parenchyma or collenchyma, which are present above and below the vascular bundle, they they extend up to the up to the upper and lower epidermis layers, and these are called bundle sheath extensions. Okay, and dear student, chloroplasts are absent in chloroplasts are absent in bundle sheath extensions. So that is about the what particularly vascular bundle. But now when we talk about the vascular bundle of xylem and phloem, you will see that in vascular bundle, specifically protoxylem, if you talk, it's situated towards upper epidermis. Okay, let's write this particular important point in the next slide. So it will be easy for you to write it. So if we talk further in this about the xylem, then in xylem, protoxylem is the first formed primary xylem, and this protoxylem is situated towards upper epidermis. Whereas if we talk about protophlloem, then this protophlloem is situated towards abaxial or surface of lower surface or lower epidermis. This way, this particular protoxylem and metaxylem are present. Apart from this, if you see the vascular bundle are surrounded by bundle sheath, and it is made up of thick walled parenchyma. Okay, dear students. So that is about the what particularly we can say about the dicot leaf anatomy. Now, apart from this, when we talk about the monocot leaf anatomy, before that, dear students, let's talk about one more important point. In dicot leaf, if you see this dicot leaf are at right angle to the what a stem. So when they are present on the light angle to the stem, upper surface receive more sunlight as compared to the lower surface. So we remember in dicot leaf are arranged at right angle to stem, at right angle to stem, and upper surface receives more sunlight, as compared to lower surface, as compared to lower surface. And so there is a difference between the internal structure of upper and lower surfaces. Okay, so this is the particularly thing about the dicot leaf. Now after this, we are going to talk about the monocot leaf anatomy. Now when we take a transverse section of monocot leaf, you will see such type of anatomy. In this anatomy, dear student, you will see that there is a adaxial epidermis, or we know that it is upper or ventral epidermis. Same way, the lower epidermis is known as abaxial or lower or dorsal epidermis. Okay. Now, apart from this, dear students, you will see the number of stomata approximately equal on both the surfaces. And between upper and lower epidermis, you will see the presence of the mesophyll cell. Here, important thing is that these mesophyll cells are not differentiated similar to the dicot and monocot leaf. This particular mesophyll cells are not differentiated into palisade and mesophyll as we have seen a differentiation in the dicot leaf. Okay. Now, apart from this, you will see the presence of xylem, that is the vascular bundle, okay, and the phloem, as here you can see. Okay. Now, after studying this particular point, actually this is not phloem. These particular cells are phloem. Actually this represents the phloem. Okay, so this way particularly these are found, and this, if we talk, these are nothing but bundle sheath extension. Okay, so this way particularly the cells are present. Okay, now this is the anatomy which you can look, which you can observe by the taking a transaction. But what are the necessary important points which we can understand from this, that we can note down? So let's note it down. So now when we take an internal structure of this dicot leaf, the important points are there. The usually Stomata usually stomata on both surfaces are equal in number. Okay. So usually the stomata on both the surfaces are equal in number. On the upper epidermis or lower epidermis, you will see the almost approximately equal number of stomata on both the surfaces. Then both the surfaces are covered by cuticle, and this thickness of cuticle is equal on both surfaces. Is equal on both surfaces. Okay, so that is the thing. Now, apart from this, if you see, then between upper and lower epidermis, you will see the presence of mesophyll cell, and we look for the mesophyll cell. These mesophyll cell are not differentiated, are not differentiated into palisade, into palisade and spongy mesophyll. Okay, in case of this isobilateral leaves, and this mesophyll cell have only few intercellular spaces, only few intercellular spaces you will see in the mesophyll cell. Okay, now if we talk about this in more detail, now in further detail we can talk about the particularly isobilateral leaf. Then you will see the two distinct patches of sclerenchyma in the form of bundle sheath extension. So bundle sheath extensions. These bundle sheath extensions, if we talk, they are two distinct patches, two separate patches of sclerenchyma present above and below, present above and below the large vascular bundle, and extend up to upper and lower epidermis layer. Okay, then they are known as what, a bundle sheath extension. So these are present of sclerenchyma, as we have discussed. In case of the dorsiventral, they are made up of parenchyma or collenchyma. Here they are made up of sclerenchyma in monocot leaf. Now, if we talk about this monocot leaf or isobilateral leaf, and when we talk about the vascular bundle, again, vascular bundle are of similar type or compared to the dicot stem. They are also conjoined, collateral, and closed. Here also protoxylem is towards upper epidermis and metaxylem towards lower epidermis. In case of vascular bundle, and the phloem is also present on the lower epidermis side, is towards lower epidermis. Now dear student, one of the important point now which we are going to discuss that actually differentiates or specifically found in monocot only, not in dicot leaf, that are bulliform cells. So first, see the location. See, the bulliform cell actually are a large sized cells which are found at this position somewhat. Okay, so these cells are known as bulliform cells or motor cells. And these bulliform cells or motor cells are exclusively found in case of this monocot leaf upper epidermis. Okay. Now, in this monocot leaf or isovalent leaf, if we talk about this bulliform cell in detail, dear student, this bulliform cell or motor cell, if we talk, they are present in certain grasses and they are present on adaxial or upper epidermis. Now, when they are present on a upper or lower upper epidermis or adaxial epidermis, you will see that these are the bulliform cells in the leaves and they are large sized empty colorless cells. They are large sized empty colorless cells and they absorb water and become swollen or turgid. So when they absorb water and become swollen or turgid, the leaf gets strengthened or straight. Leaf become straight, or we can say they unroll. So the leaf surface get exposed. So what this statement is saying, if these bulliform cells are rich in water, they are applying the pressure sideways on the epidermal cell. So leaf is going to be flattened. The leaf on this particularly will look like this if the bulliform cells are like this. But okay, the leaf will be exposed like this. Okay. But if the water loss occur and this become fleshy, they make leaf to curl inward. They make leaf to curl inward to minimize water loss. That is the important thing with respect to this. So in this case, in second case, they will curl like this. Okay, so they are not going to be strengthened. So this is in case this in this case, and in this case, this condition is going to be observed. Okay, so this is all about the bulliform cell and all about the isobilateral leaf or the monocot leaf. I hope now you are able to understand the differences between dicot leaf and monocot leaf. These Gulligram cells are exclusively found in monocotyledon and the upper epidermis. When we compare these two, in these two, the mesophyll cell difference is one of the important point that in dicotyledon we have studied that mesophyll cell are differentiated and they are of two types, that is palisade mesophyll and the spongy mesophyll. But in monocotyledon for isobilateral they are of only not they are not differentiated, they are of only one kind. Apart from this, we have discussed that particularly the number of stomata are more on the lower epidermis in dicot sting, but in isobilateral or we can say the monocotyledon, the number of stomata are equal on both the surfaces. Okay, so these are the some of the differences which help us to identify. And obviously, remember that in particularly uh, we can say dicot leaf mainly you will find a reticulate venation, and in veins this particular vascular bundle are found. And in every leaf, the veins will actually particularly locate this vascular bundle. Now, in case of isobilateral leaf, if we talk the arrangement of leaf, you will see that the leaf are somewhat arranged parallel to the stem. Leaf are arranged parallel to stem in monocot leaf. So when they are arranged parallel to the particularly stem, so both surface of leaf receive equal amount of sunlight, equal amount of sunlight. So no difference occur between, so no difference occur between internal structure of upper and lower surfaces. Okay, I hope you understood, dear students, and this is found in case of the monocot. Now, one more thing which we can compare between the dicot leaf and monocot leaf, that the size of vascular bundle is dependent on the size of veins. Actually, veins vary in thickness in the reticulate venation in dicot leaf. So the diameter or thickness of veins will also get vary. So this is we talk about the one comparative difference between these two. In dicot leaf, if we talk, the thickness of veins, thickness of vascular bundle, or we can say size of vascular bundle. In dicot leaf, size of vascular bundle vary from one vascular bundle to another vascular bundle because thickness of veins is different in a reticulate venation. Why? Because this vascular bundle are located within the veins. So we can say the size of vascular bundle, size of vascular bundle are dependent on size of veins. But when we talk about the monocot leaf, here also size of vascular bundle dependent on thickness of veins. Here the size of vascular size of vascular bundle is nearly similar is nearly similar except in main vein because they have what have parallel venation, and due to parallel venation the size of the vascular bundle is going to nearly similar because the size of the veins is going to be almost similar. So these are some of the differences which help to identify or differentiate monocot leaf from the dicot leaf. Now let's discuss some of the questions. In dicot leaf, the tissue between upper and lower epidermis is called, obviously the tissues which are present between the upper and lower epidermis which represent the ground tissue, they are nothing but a mesophyll cell. So the fourth is going to be the correct answer. Mesophyll is made up of, obviously the are made up of parenchymatous cells. Okay. Now the size of the vascular bundle is dependent on, obviously the size of vascular bundle is dependent on the size of veins. In reticulate relation or dicot leaf it is variable, and in case of monocot leaf or isobilateral leaf it is a similar, nearly similar. Mesophyll is not differentiated into palisade and spongy parenchyma in. Obviously, it is not represented differentiated in case of the monocot leaf. We have discussed this. Okay, dear students. So this is all about what a dicot leaf and monocot leaf internal structure anatomy. I hope you understood well. Thank you.